Hey everybody, welcome to our November 19th PacVec seminar. Today we have two great speakers. The first is going to be Ben Nyman from UC Riverside in Dr. Karthik Chandrasargarian's lab. So Ben is broadly interested in insect ecology and how habitats shape vector populations and interact with vector control to impact downstream public health consequences. During his dissertation work, he grew passionate about using advanced modeling methods to garner new insights from all ranges of vector data, from individual larval habitats to multi-species landscapes. He's also interested in developing new analytical tools for improving efficiency and value of public health data in integrated vector management programs. With that, Ben, I'll pass it over to you. Please share your slides.
All right. Thank you for reading my word salad. That
It
was
was great.
awesome. All right. So, hi everyone. I'm Ben, and today I'm a postdoc in Dr. Chandrasekharan's lab at UC Riverside, who's a new faculty member in mosquito ecology. And my talk today is going to be about modeling environmental impacts on Aedes communities across Southern California using surveillance data. So let's see. There we go. So as you'll hear today, for both me and Zach, I'm pretty sure, surveillance is a vital and powerful tool in vector management and research. It's maybe the most important tool in integrated vector management because it allows us to do many things. It allows us to monitor the presence and movement patterns of vector species. We can also use surveillance to detect and monitor pathogens of importance. And then from there, we can also use this data to plan interventions like abundance-based treatment thresholds or public health interventions if we detect a pathogen somewhere. This surveillance data also gives us a ton of really rich data, both spatial and temporal, much of which is very standardized by different traps and trapping methods we use. And we can use that data to better understand the ecology and biology of the vectors, which in turn can then kind of feed back into our surveillance programs and integrated vector management to create this like positive feedback loop of more efficient and effective vector management. So in Southern California, our main, domestic mosquito was Culex quinx, which is kind of docile in some relative respects. It's crepuscular. It needs kind of large bodies of water to lay its egg rafts, or relatively large, and it is one of the primary West Nile virus vectors in the U.S. But in the last 10 years or 15 years going on now, we've got the introduction of many invasive Aedes species. So Aedes albopictus, Aedes aegypti, and Aedes nondescriptus, all three of which have kind of spread out over the landscape and are currently shifting and changing their distributions. They differ from Culex in a lot of ways. They require very small amounts of water. They're diurnal. They love biting people and living around our houses. And it kind of makes us miss, those good old days where it was just kind of Culex quinx around. But anyway, even though they're ostensibly quite similar in many respects, our three, in this case, I'll just be talking about two Aedes species, can differ in a lot of ways. They can have ecological differences in relation to their habitats or niches. They can have behavioral differences in terms of biting preferences or nectar utilization. And they can even have important vectorial capacity differences down the line. And if you're interested in pathogens, you probably will enjoy Zach's talk after this one, but we will no longer be talking about pathogens. So my question for this presentation was really, towards the end of my dissertation, I got into a lot of surveillance data, and I was interested in how surveillance data can inform us about the interactions between the mosquito community and the Southern California landscape. And in this case, this is a very limited community. We're not talking about, these natural worlds of mosquito diversity. We're really just talking about a few species. So to conduct this research, I wanted an area that had multiple invasive Aedes species. So I worked with San Gabriel Valley Mosquito and Vector Control District in the West San Gabriel Valley, so the whole San Gabriel Valley is kind of the orange region, and the green square is kind of the West San Gabriel Valley where the trapping was conducted. So within this region, we established 12 trapping sites between me and the district. So some were operated by me and some were the district personnel. And we trapped at each of these sites for 24 hours, once a week for for 8 weeks during the mosquito peak season in the fall. And this was done using BG Sentinel-2 traps baited with the BG lure. So from this, we collected 6 different species, but we'll focus mainly on the 3 most abundant species, so Aedes aegypti, Aedes albopictus, and Culex quinx for the rest of the presentation. So across our 12 sites, we got different numbers of mosquitoes, and also the proportions of the species we collected varied across site. And this kind of brings up a lot of interesting questions about what about these sites are dictating these proportions of mosquitoes collected? And these sites, varied a lot. We had some sites that were, people's backyards in a very suburban area. We had public parks. We had the LA Arboretum as one of the sites. So there's a lot of variation, and there's also a lot of variation in this landscape. So at the bottom left of this, just to orient you, we have you're getting into LA proper. So that's like urban sprawl, residential sprawl LA. And then in the north, both the northwest, we have the Verdugo Mountains, and the northeast is the San Gabriel Mountains. So a much more natural foothills area. So to characterize these trapping sites and get information about how that relates to the mosquito community, I used lots of publicly available data. So in like 3 broad categories. First, climate. So historical climate, as you can see, this top left box, that's kind of like the average mean temperature, which does kind of get cooler as you move away from LA proper. I also wanted to look at urbanization and land use. So I have national land cover database data as well as impervious surfaces. And then we also looked at vegetation, so tree canopy cover and vegetation index. So if you take all this data and you have all this occurrence data and all this environmental data, you can kind of begin to venture into the world of species distribution modeling or niche modeling. And that's where you use the patterns of species occurrences and patterns in the environmental data to create a model that can predict whether or not a species will be present in a novel area or predict the abundance in a novel area. So we have our first step done here, which is just to collect occurrence data and environmental characteristics. Then we would need to fit some sort of predictive model, and then we can apply a novel scenario or novel data to the model. So a greater spatial extent, interpolation between trapping sites, or even like a temporal shift of like a climate change scenario. However, when it comes to predicting a model, you need to select a response variable. And in this case, we're kind of challenged by a small spatial scale. So if we're talking about a single district in Southern California, many of them aren't trapping on a large scale with a huge number of traps. So if we look at presence absence, I mean, in all 12 of our sites, we got some amount of Aedes aegypti and Culex quinx and a varying amount of Aedes albopictus. So we can't really use presence absence. Presence only relies on pseudo absences where you kind of take nearby points that you didn't trap at and assume that it's not a species isn't found there, which I don't think would be very appropriate for this data because the mosquitoes are there. And then the last one's abundance data, which could work for this because we did get some gradients of abundances. However, you kind of get into a bit more computationally difficult space, and we'll kind of address that later. But to avoid all these pitfalls, I looked at this from a trap rate perspective, where you have a BG Sentinel trap and you collect X number of mosquitoes. What proportion of those mosquitoes might you expect to be Aedes? What proportion might you expect to be Culex? And from that, you can kind of glean some information about the habitat suitability for each of these species because that proportion varied across the landscape. And we can also use this framework to analyze, differences between Aedes aegypti habitat and Aedes albopictus habitat. So getting into that, what we can do is kind of on the y-axis here, look at our probability of detecting Aedes. And on the x-axis, we can assign some environmental variable. In this case, this is a principal component of the climate. And we can see that there is this kind of univariate response to climate where, as this principal component increases, our probability of detecting Aedes over Culex also increases. And we can extrapolate that out to a larger model where we have climate, land use, excuse me, and vegetation. And we can see all of these things significantly correlate with our presence of Aedes. So on this tree plot, as these odds ratio estimates move towards the right, you're predicting more Aedes, and as they move towards the left, they're predicting more Culex, with the stars denoting the level of significance. And all three of our land use categories kind of significantly influence this. And if we look at the predictive power of this model, it's pretty good. We can see there's separation between the Culex predictions and the Aedes predictions, as well as an AUC of about 0.8. If we take the same framework and apply it to our two different species, the model performs a lot worse because the range of Aedes albopictus collections was a lot more limited than the overall Aedes versus Culex, but we could still detect some responses here. And looking at that same model for this trap rate comparison with Aedes aegypti collection proportion on the right and albopictus on the left, we do see that there is some significant contribution of climate and vegetation, but all of our land use characteristics were not significant, and this model did perform worse, as you can see from here, where the best this model can do is distinguish between Aedes aegypti habitat and habitat that's about 50-50 Aedes aegypti and Aedes albopictus. So we looked at these logistic responses to the variables, but you might expect that a lot of these interactions might be a lot more complicated. So we want to apply something a bit more robust. So I looked towards boosted regression trees. So this is a classification model that uses machine learning to build up classification trees and kind of average out the predictions, which lends them a lot of accuracy and high predictive performance. And this is just an example of, if you survived the Titanic or not, based on, your gender, your age, how many family members you have. So incorporating a lot of different variables in a lot of different ways, and then iterating through lots of trees in order to find what is actually important. So if we compute that for our Aedes versus Culex model, we get, these are our top four contributing variables, which is a climate principal component, temperature, surface impermeability, so that's kind of related to urbanization, and precipitation seasonality. And if you can see, they have like a low value, that's kind of one prediction. And then as you move down the x-axis, which is our environmental variable, there's a threshold where you switch to the other prediction. So those are the kind of the nodes of the tree as they appear in each of these models. And if you look at the predictive performance of this model, it does better than the GLM. And you can see more separation between the regions that were predicted for Culex versus Aedes, and the AUC is also higher. If we do the same thing for the Aedes aegypti versus Aedes albopictus, it's a bit more iffy. We have different highest contributing variables. And if you look at the whole model, it actually is pretty similar. And actually the AUC is worse. I think this has to do with when you break down the data for cross-validation and you have a low sample size, it doesn't really perform very well. But that's fine. What we can do now is take these outputs and map them across the landscape. So if we look at our Aedes versus Culex boosted regression tree model across the landscape, we can identify regions where we have higher predicted Aedes trap rate versus Culex. And it kind of partitions out to the whole southwest region, kind of Monterey Park down to East LA, being very low prediction for Aedes, which is questionable. And if we apply our collection data on top of this, we can see that there is some kind of interesting incongruity with what you might expect. But that's definitely something that is interesting to look at. If we look at our species model, so this is Aedes aegypti versus Aedes albopictus, with redder being Aedes aegypti prediction, and bluer, if you look at the the legend on the right, it only goes down to 0.5, so that means 50-50 split. So the blue areas aren't albopictus dominated, they're predicted to be about 50-50, and that's just because of the input data. And we see that, as you move towards East LA in the southwest area of this map, you get much higher prediction for Aedes aegypti. And then as you move up into the foothills of the St. Gabriel Mountains and other mountains, you get more albopictus. So, and this is just showing the species breakdowns from each of our trapping sites and how they correlate. So here you can actually see the pattern of the more northern sites having more albopictus, and that being reflected in the predictions on this map. So using this framework, we were able to see that there is a strong environmental correlation where you can kind of see the interaction between the environment and the Aedes versus Culex community. So you can actually predict Aedes using a lot of different factors. However, our ability to predict habitat for Aedes aegypti versus Aedes albopictus was strongly curtailed by our lack of good Aedes albopictus data. So that's a bit more questionable. However, in this entire system, you have a lot more going on than just the environment influencing the species. You have the species influencing each other as they compete for larval habitat and nectar resources. And then you also have the environment mediating those interspecific interactions. So as a kind of a final part to this presentation, to look at this, I applied a joint species distribution model. So this is a much more complicated model that includes the environment's effect on each species, but also each species' interaction on each other and the environment's effect on the interactions between each species, as well as lots of random structure. So I used a program called HMSC, and this is a Gibbs sampling Markov chain Monte Carlo estimation that can accommodate both presence, absence, or abundance data. So here is where I kind of leveraged that abundance data in this model. And what we can see is that we can look at how much variance is explained by each of our environmental variables for each species in the entire model. And we see that Aedes aegypti performs the best. And interestingly, Aedes albopictus performed better than Culex Quinx, even though there's a lot less input data. But I think that's because Culex Quinx had a lot less variation over the environment. So it was less, it was harder to predict how it would respond. We can also look at something called a beta plot in this program, which is just each species' response to the environmental variables we use. So I'm not going to go into all the details here, but we recovered a lot of the important determining variables from our models, our previous models, like impervious surfaces, precipitation variation, and social vulnerability, as well as other aspects. And then also important, we can look at interactions between species. So this is a residual correlation matrix where we can look at the abundances of each species in relation to the presence of another. So if we think about classical competitive exclusion, things like that, where you have a projected niche, but a species isn't present, but another species is, you might expect them to be being excluded somehow by a superior competitor. And that would show as blue or a negative value on this matrix, but we actually see all positive trending interactions and even a significantly positive interaction between our two Aedes species. So not only are they not competing, seemingly the presence of one is a positive indicator for the presence of another. And looking at those posterior distributions of abundance predictions, this is just one example of what that looks like after you compute it, the MCMC. So here we have social vulnerabilities impact on the Aedes aegypti abundance. And if you add all these together, then you can output a distribution map like this. And this is our Aedes aegypti abundance prediction based on our JSDM. And you can see it actually looks pretty different than the species prediction map from the boosted regression tree. We have specifically very high prediction as we move closer to LA in the bottom left, low prediction towards this bottom right corner, And then actually going back to higher predictions in the natural areas in the foothills of these northern mountains, which is interesting. And again, we compare it to our input data, and it's kind of difficult to interpret, but we'll get back to this in a second. Looking at our Aedes albopictus abundance in space with predictions, we have a similar pattern to Aedes aegypti. However, there's some notable differences. So there's less predicted abundance in the southwest LA region and higher predicted abundance in the foothills of the San Gabriel Mountains, which does agree with our previous species trap rate models. And then interestingly, if you take both of these predictions and stack them on top of each other and color code them and change the habitat suitability to opacity, you can get something like this, where we have red areas being higher prediction for Aegypti, blue areas for Albopictus, and mauve kind of being predictions of both. And to kind of sanity check this, I overlaid the iNaturalist data from the last 5 years for both of these species. And you can actually see that there does seem to be a decent amount of agreement in terms of where people are encountering Aedes albopictus and Aedes aegypti, although this is a very limited data set. So, go out there, and if you work on mosquitoes, find your mosquitoes and upload them to iNaturalist, please, so we can do more analysis on something like this. And if you compare this, the iNaturalist data to the boosted regression tree, this one actually has a lot more incongruity. So it does kind of bring into question how reliable something like this would be for the amount of data that we inputted. So in conclusion, there's a lot of ways we can evaluate environmental effects on mosquito communities, and using surveillance data for this is valuable, but from a single district or single trapping seasons, a lot of this data is unsuited for normal SDM frameworks. However, binary trap rates will allow for analysis of hotspots or niche partitioning while accounting for bias and trapping at a low rate. And then further down the line, SDM and joint SDMs can provide insights into current and future populations. As another consideration, JSDM is a lot more complicated to implement and a lot more computationally taxing. It's like the boosted regression tree can run in like a few seconds, and the JSDM for this limited data set took like a 24-hour period to compute. So just as a comparison. In the future, I like to continue looking at these comparisons of models using more quantitative measures and more data sets to kind of compare, when can you use certain modeling frameworks compared to others. I'm also looking at optimizing trap site selection for modeling vector populations. So if you have, more sites than you can trap at, please get in contact with me. I want to discuss with you how you might be able to choose which ones are the best for that. And I'm also looking to do further JSDM, including areas that have more Aedes nodoscriptus to kind of get a hold of what its niche is. And with that, I'd like to thank the land that this work was conducted on, as well as PacVec, UCR, all the people that let me trap, and San Gabriel Valley Mosquito and Vector Control District, as well as my committee and collaborators, and I'll take any questions.
Oops. All right. Thank you, Ben. Let's see. Does anybody have any questions to start us off? There we go. We've got a question from Kristen. Have you published any of this yet?
So the answer is no. So this was my dissertation. Well, I guess in my dissertation, yes, but for the paper, we're still looking at more quantitative comparisons of the model output. So that's what the publication will be centered on, and that should be out next year.
Thank you. Looks like Chris has a question.
Yeah, I was just curious how you reconcile the suitability estimates with the origins of Aedes albopictus in particular in your study area, which is actually around El Monte and South El Monte, which looked like the lowest suitability places on the map.
Yeah. There's a lot of discussion about what are the niches for these species. And for me, what I've observed in Southern California is definitely that Aedes albopictus is usually more associated with the rural to suburban interface or the natural area interfaces than Aedes aegypti. And we do see a lot of both species in El Monte. We didn't have any trapping sites that were like dominated by Aedes albopictus, and that's kind of reflected in the El Monte results. But then even like in the predictions from the joint species distribution model that have like really high prediction in like Pasadena, again, we do detect a lot of Aedes albopictus, but there's no like dominance of areas that have dominance of Aedes albopictus compared to Aedes aegypti, whereas we do see a lot of areas that you only detect Aedes aegypti, and those are like the most heavily urbanized areas. So yeah, I'm interested in also like considering how this compares to like Florida and other areas where they have come into contact, and we're seeing different outcomes.
I know you're limited a bit by the scale of the data, but if you could pick an optimal scale for predicting the distributions of these species, what would that be?
Um, that's interesting. There's some studies talking about incorporating high density, small scale trapping into larger data sets, using things, using like much more large scale modeling, things like Maxent, which are interesting to me. But I do think it kind of depends on what the question you're asking is, and what can you capture in your environmental variables or your characterization of the environment. So at this scale, I think it's I would want to go a little bit bigger just in order to kind of control for all the variation and land use within a single category that can be very variable. But I wouldn't say that this would be applicable for like an entire US model. I think there's a lot going on on the East Coast that just isn't congruent with this modeling. And there's like some interesting over applications, like Lindsay Campbell did like an Aedes nodoscriptus model in like 2016, and they in Australia, and they projected it to California because we were just detecting it here, and it was like all in like the Central Valley and then like Bakersfield. So there's a lot of interesting incongruities with just like the scale. But I think like just kind of the greater LA area would kind of be my ideal range for this.
Thank you.
So Hongwei asks, how often do you trap all three kinds? If you're asking about Culex, Aedes aegypti, and Aedes albopictus, it was often. So any site that we collected Aedes albopictus, we collected all three. So that is pretty often. So were my AUC values measured in bag or out of bag? So it depends on the model. So like the GLM, it was, I think I'm correct in saying that it's in the bag because we just ran the data back through the model. Whereas for the boosted regression trees, there's a cross-validation occurring, but none of it was in, so I guess is a cross-validation out of the bag? I don't know. It's not new data, but it's data that's kept out of the model and then tried. So you can glean the answer based on that, but I'm not sure. Could the model incorporate continuous trapping data like a BG counter? Sure. Both of these methods are kind of robust to inclusion of data. You might have to change how you analyze things. So in considering like temporal or spatial random effects might be important. And also an important thing is I didn't really consider weather in this case because I was looking at the proportion. Whereas if you're looking at, abundances over time, weather is going to be really important for that. So there's a lot that would have to go into it.
Okay, thank you. And then, so Matthew Lopez asked if we'll be able to access the recording for the meeting. And of course, Chris put that the link for the recordings are on the PacVec page. So you can snag that link in the chat. Ben, thank you so much for your presentation. It was great. We had really good questions, and I think we will move along to Zach next.
Thank you. Let me stop sharing.
All right, Zach, would you like to put up your slides, please?
Yeah, I can do that.
Okay, looks good. Okay, so for our second talk, we're going to be hearing from Zach Barrand, who is a PhD candidate student at Northern Arizona University and a research associate at the Pathogen Microbiome Division of TGen North. He's in my lab. So he's gotten his Bachelor's of Science in Microbiology from NAU, research emphasis in the phylogenetic diversity of tick-borne co-infections. And so the research he's going to be presenting today, while my group is funded under PacVec for West Nile virus, we also have a tick-based project going on as well. And Zach is going to be presenting on that. So Zach, take it away.
Great. Thank you, Crystal. So I'm going to be talking about the work that our group is doing to track spotted fever Rickettsia through Northern Arizona. And so my talk today is going to be giving a brief background on tick-borne diseases. And then I'm going to jump into Rocky Mountain spotted fever in Arizona, and then focus the rest of the talk on the work that we're doing for spotted fever Rickettsia surveillance in Northern Arizona. And so tick-borne disease is distributed across the entire contiguous United States, with a heavy concentration within the Eastern United States and specifically the Northeastern United States, with 6 major diseases, including Lyme disease, ehrlichiosis, Rocky Mountain spotted fever, babesiosis, anaplasmosis, and tularemia. And we can see within the Western United States, we have a more sparse distribution within reported tick-borne disease cases. However, we do have them, including Rocky Mountain spotted fever. And of course, disease presence is dependent on vector presence. And these vectors include a few medically important hard ticks, such as Haemophilus species right here, Amblyomma species, Rhipicephalus species, Dermacenter species, and Ixodes species. And Ixodes being well known for being the vector of the Lyme agent Borrelia burgdorferi, where all of these medically important hard ticks can transmit various pathogens. And kind of going over a basic life cycle of a hard tick, this is going to be the life cycle of Ixodes scapularis. That's going to be the deer tick in the Northeast. We can see on this life cycle plot here, there's going to be 4 major life stages, and that's going to be egg, larva, nymph, and adult. And the 3 life stages of following hatching of the eggs are going to be associated with a blood meal, where these ticks can either acquire or transmit a pathogen. So now let's move on to Rocky Mountain spotted fever within Arizona. So Rocky Mountain spotted fever is a bacterial pathogen that is caused by the bacteria Rickettsia, and it is vectored by a few tick species: the brown dog tick, Rhipicephalus sanguineus, the American dog tick, Dermacenter variabilis, and the Rocky Mountain wood tick, Dermacenter andersoni. And we can look on this map here of spotted fever rickettsioses cases reported from 2000 to 2013. And we can see that there is a heavy concentration of spotted fever rickettsiosis within the southeastern United States. But I want to draw your attention back to the West Coast or Western United States over here, where we still see it in pretty high reported numbers. And specifically within Arizona, we have almost all the counties in Arizona reporting human cases, with a heavy concentration within Navajo County and Gila County. And when humans do get infected with Rickettsia rickettsii, symptoms can present with fever, headache, this macular papular rash, nausea, vomiting, stomach pain, several other symptoms, including long-term problems such as blood vessel damage and also CNS disease. And then the disease can quickly progress to life-threatening if left untreated, but it is treatable with doxycycline. However, Rickettsia rickettsii is not the only known rickettsia that can cause spotted fever-like disease. And so looking at this phylogenetic tree here, we see there's a lot of species that are present within the spotted fever group of Rickettsia. And if we look even closer here, all of the Rickettsia with a star next to them are known to cause human disease, and the ones in bold have had associated CNS disease cases. And so we can see there's quite a few spotted fever-like pathogens within the Rickettsia genus. And this includes Rickettsia parkeri, which we have seen in Arizona, as well as some others like Rickettsia africae and Rickettsia macillae is also going to be one of those. With the vector species ranging from the dermacentor species that I've already mentioned, as well as Amblyomma maculatum and Rhipicephalus sanguineus. So if we look at the Rocky Mountain spotted fever cases within Arizona, the first detection of Rocky Mountain spotted fever was in 2003. And since then, we have seen an increase in cases since that initial detection, as well as consistent cases year after year of Rocky Mountain spotted fever. And these cases are going to primarily be in Arizona tribal communities with free roaming dogs. And in those communities, vulnerable groups such as children, elderly, and those with weakened immune systems are going to be who are impacted. And roughly 10% of RMSF cases in Arizona are fatal. And in Arizona, our primary tick vector for Rocky Mountain spotted fever is going to be the brown dog tick, or Rhipicephalus sanguineus. And this tick is going to be the primary vector within Arizona as well as northern Mexico, which is slightly different ecology than what is observed in the rest of the United States. And so within this transmission cycle, with Rhipicephalus sanguineus being the primary vector, it's going to be roughly the same cycle that I touched on briefly at the beginning of this presentation. However, it's going to be between the brown dog tick and dogs. And so we're going to have those four major life stages. We're going to have egg, larva, nymph, and adult, where the larva, nymph, and adult are take a blood meal. And it is during these blood meals that the tick can acquire or transmit Rickettsia rickettsii. In addition to the transmission between dog and tick, larvae also have the potential to transmit Rickettsia rickettsii, since transovarial transmission is possible from adult female to the eggs that they're laying. And so humans can also be bit at any point during this life cycle and transmit Rickettsia rickettsii. So now let's get into the goals and what we set out to do. So we wanted to first expand tick surveillance efforts within Arizona as a whole. And we wanted to focus specifically on identifying brown dog tick distributions within northern Arizona. And we wanted to understand the prevalence of the different spotted fever group Rickettsia that were present within these populations. And so to do this, we collaborated with High Country Humane and Coconino Humane Association to collect ticks off of dogs on intake. And these dogs came from various locations throughout the northern Arizona. The ticks were then brought back into our labs at TGen, and we identified them to life stage, sex, and species based off their morphological characteristics. And looking at what we got off of the dogs, we got a total of 330 ticks off of 30 dogs, with the majority of these ticks being adults, with 77.2% of the ticks that we collected. And 21.6% of those were nymphs. And if we look at in our adult tick population that we got, we were, the majority of our females are engorged, whereas our males are not. That could be that's most likely due to that males don't engorge, and so because they don't have a soft abdomen, and so it's a lot harder to detect a blood meal within the males just off of physical observation. We then took the 330 ticks and pooled them into 150 pools. The pools were constructed with a single female per pool, up to 5 males per pool, and up to 50 larvae or nymph per pool. And we didn't mix ticks from different dogs, so keeping those distinct. We then extracted the tick pools and we ran a pan-Rickettsia qPCR that detects the presence of all Rickettsia. So that's going to be pathogenic and non-pathogenic Rickettsia species. And looking at this amplification plot, we're going to have this threshold line right here in the middle that the arrow is pointing to. And all of these amplification curves are going to be crossing that threshold when there is a fluorescence of the presence of any kind of Rickettsia. Any amplification curve that crosses that threshold before the 40th cycle of the PCR will be deemed positive. And so we got 20% of our samples were positive for the presence of any kind of Rickettsia. So then we took the samples and passed those through a Rickettsia qPCR, so looking for the agent of Rocky Mountain spotted fever. And we found the presence of no Rickettsia within these samples. But we were still curious what's the Rickettsia that we are hitting to that we detected within that pan-Rickettsia qPCR. And so we took two different approaches. And so we looked for the presence of a spotted fever group gene, so that would be able to detect the presence of any spotted fever group Rickettsia. And then we also went after bacterial 16S, which is going to be detecting the presence of any bacteria within those samples. And so we ran the PCR and ran a PCR gel. This gel is going to, we load our samples in with a known DNA ladder, and it's going to basically organize those by DNA fragment size, starting down here, where the smaller the DNA fragment, the faster it will run through. So the lower it will be in the gel. And so at the bottom of this ladder, we have 100 base pairs, and it goes up to, our target is at 700 here for the spotted fever group. And we can see that a few of our samples have some bright banding. And so we had 9 out of the 30 samples that were positive for pan-Rickettsia came up for the spotted fever group Rickettsia PCR. And looking at the bacterial 16S, 100% of our samples that came up for pan-Rickettsia, so all 30, amplified for the bacterial 16S. We then took the samples that were positive for spotted fever group Rickettsia and bacterial 16S and sequenced those on Illumina sequencing. And then we took the resulting sequence reads and went and classified them for what bacteria was present. And so looking at our bacterial 16S results for one of our samples, this is going to be a flow chart that came out of the bacterial classification. And at the bottom here is going to be labeling the taxonomic level in which the reads were classified. And each bar is going to be representing how many reads were within that taxonomic level. And so down at the bottom, that's going to be domain, phylum, class, order, family, genus, and species. We had a total of 160,000 reads that were classified to bacteria. And of those, 66.2% were classified to the genus Rickettsia. And this sample specifically is a pool of 5 males collected off a dog in Flagstaff, Arizona. And so if we look at the spotted fever group bacterial classification on the same sample, we can see that we had 167,000 reads classified to bacteria. And we got 100% of those classified to Rickettsia, but we also got a species classification where we got 38.5% of those to Rickettsia maciliae, a spotted fever group Rickettsia. And so, and if we look at this same sample on our pan-Rickettsia amplification plot, this was our highest concentrated sample where it had the lowest cycle threshold value. So it crossed very early on in that PCR cycling. And if we look, and across all of our samples, we had a total out of 3 total samples out of the 30 that we tested for pan-Rickettsia had reads classified Rickettsia maciliae. two of which were off of the same dog. And so in addition to this pool of 5 males, we had an additional pool of a single female come off the same dog that was also positive for Rickettsia bacilliae, leading us to believe that that dog was positive for Rickettsia bacilliae and transmitted the pathogen to those ticks. So now, what is Rickettsia bacilliae? And so Rickettsia bacilliae was first isolated from Rhipicephalus sanguineus in southern France in 1992. And it has been known to cause spotted fever-like disease in Europe, specifically Mediterranean-like spotted fever. It can be treated with doxycycline like Rickettsia rickettsii. However, Rickettsia bacilliae is resistant to rifampin. And in the United States, the last published detection in brown dog ticks was in eastern Arizona in 2004. And this organism is vastly understudied when compared to Rickettsia rickettsii. And so we wanted to have a better idea of what was the pathogenicity of Rickettsia bacilliae in Arizona. And so we started by obtaining a full genome of Rickettsia bacilliae out of that pool of 5 male ticks. And we constructed this maximum parsimony tree that we see here. And we have both the Arizona genomes in there, so our genome, as well as that one from that earlier in the 2000s. And then we have the Southern France isolate as well. And we see that there are over 7,000 nucleotide changes from the Southern France isolate to the Arizona isolates. And so they're very different from one another. And so we needed to do additional studies to better understand how infectious Rickettsia bacilliae is here in Arizona. In addition to this, we want to perform more robust tick surveillance to identify what other spotted fever group Rickettsia are within northern Arizona, as well as try to understand more about Rickettsia bacilliae to figure out what is the transmission cycle that is occurring. Is it the same as Rickettsia rickettsii, or is it different in any way? As well as other tick-borne pathogens such as anaplasma or lichiosis and some others. As well as we need better diagnostics to cater to the pathogenic Rickettsia present in northern Arizona, since what is currently being done is serology testing, and there is cross-reactivity occurring. And so we're not sure what actual rickettsiosis are impacting our tribal communities. And I want to acknowledge, with that, I would like to acknowledge our team here at TGen North, specifically Alexis Rivas and Naja Jones, as well as Crystal Hepp, and our collaborations with High Country Humane and Coconut Humane Association. And before I take any questions, I want to just acknowledge TGen will be hosting the 6th annual Southwest One Health Symposium on December 11th at Arizona State University in the Memorial Union. And there is a QR code there for registration. The symposium is going to be from 8 to 4 p.m. on the 11th, and then ending with the Nathan C. Nieto One Health Memorial Lecture from 5 to 7 p.m. I can take any questions.
All right. Great job, Zach. Thanks. Okay. Does anybody have questions? It looks like we have from Scott Baker. Were any additional French isolates used in the genome comparison between the Arizona isolates and the original French isolate of R. bacilliae?
So no, so the only genomes that were used in that comparison are the ones that are listed, mainly because those are the only genomes that are available. So we need to collect more genomes to have a better idea of all of that. It is not only understudied within the US, but around the world. There are only 3 known human cases to date, 2 of which are in Europe and 1 is from Argentina.
Fantastic answer, Scott says. That's really nice. So next, Christian says, great study. Did the other Rickettsia positive tick pools have non-pathogenic Rickettsia?
Yes. So we need to look into that a little bit more, but they were predominantly so Rickettsia ripsefali is going to be what dominated those pools, and that is a spotted fever group Rickettsia. So it would come up on that spotted fever um, assay that we ran, but it is non-pathogenic.
Okay. Oh, Chris has got a question.
Yeah, I was just wondering if there's anything general you can make of the phylogenetic structure within the spotted fever group Rickettsia, because I noticed Parker I, for example, is in there with Rickettsia rickettsii in the same clade. And then Maciliae falls out in a separate clade. And I know these are closely related, but is there anything general in terms of pathogenicity or other phenotypic characters for those?
That's a great question. So yeah, so as like what Chris is saying, so we can see that Rickettsia rickettsii and Rickettsia parkeri are more closely related to one another, whereas Rickettsia maciliae is kind of out here on its own. So yes, so Rickettsia rickettsii is associated more with severe disease within humans than what has been observed within Rickettsia maciliae. And so that would be kind of phenotypic differences, like in terms of like what we're seeing when it presents within a clinical case where it's not causing as severe disease.
Zach, do you remember at the RMSF meeting that just happened in Arizona recently, up here in Flagstaff, do you remember what the CEC said about the number of RMSF cases that were actually confirmed to be Rickettsia rickettsii?
I don't remember the exact number, but I remember that the PCR validation of the serological or the serotype or the serology is very low. Do you remember the exact number, Crystal?
It's 2%.
Okay.
So I think that I think that we probably don't have a super strong understanding of. It's possible that Rickettsia rickettsii is causing the vast majority of these, but with only 2% validation, I think it's probably worthwhile for us to be looking at other spotted fever group Rickettsia as potential causative agents.
Thank you.
All right. Oh, it looks like so there was also Rickettsia maciliae found in Derma Center in the inland northwest. Very cool. Let's see. I'm not sure if I'm saying your name right. I'm sorry. Christian, did you end up sequencing?
So I need to correct that. That's not what I meant. What I meant is that we found a lot of Riverside in Derma Center. We were looking for Rickettsia, and we just couldn't find it, but we found a lot of ticks with the non-pathogenic one. So
And
yeah.
what was your approach?
Oh, we were just using single PCR, one by one. A lot of work, not much result, but that's how it was. I had another question actually. I was reading about this east side hypothesis that ticks that have the non-pathogenic Rickettsia are less likely to have pathogenic Rickettsia because they sort of have this immunity. Is that hypothesis still standing, or am I completely, misled by that?
So the hypothesis that the presence of non-pathogenic is kind of taking up where pathogenic could be, right?
Yeah.
That's okay. I think so. I would need to look into that a little bit more in terms of how would fit into, say, our system in Arizona. But I think when there is a Rickettsia or any kind of bacteria filling one niche, it might not necessarily allow for, say, pathogenic to take hold. And I think that's definitely something that we've talked about and hypothesized is occurring within tick populations here in Arizona.
I remember reading about it in Montana back in the days.
Yeah, I think that's really exciting, though. I mean, it would be sort of a great thing to understand better. So it looks like Tara Roth asked, have you detected any evidence of co-infection of Rickettsia?
I don't think we have yet. So I think we are seeing the dominance of certain Rickettsia within certain ticks. However, our approach, I think we could, looking at our, let's jump over to our flow chart here. So looking at this, we can see within the bacterial success, we're not necessarily getting any species level information here. And then when we jump over to spotted fever group, we only have a small portion of those actually being classified predominantly to Rickettsia maciliae. These classifications are influenced by our genes are relatively large that we targeted, and we had to fragment those to sequence them. And so when we put them through our analysis, we didn't reassemble those. And so we're going to have some reads that drop out and don't get classified. And so we could clean up our analysis and kind of see if we actually do have co-infection of, say, pathogenic and non-pathogenic or other non-pathogenic Rickettsia together through assembling our reads first and then passing them through our bacterial classification.
Zach, since it's an entire 16S amplicon, what's another approach that you could take? Since it's a long amplicon.
Oh, yeah. That took me a second to realize it. So on our methods, we have been successfully sequencing our larger amplicons on PacBio sequencing. So rather than fragmenting those, we are keeping them together and passing them through long read sequencing. And so we keep those long amplicons together, and then we would get higher resolution. Especially on that full gene 16S, we'd be able to get species level information out of that.
Okay. It looks like we have covered all the questions. And so like Chris put earlier, of course, the recording can be found on the PacVec website after you scroll down a little bit under the seminar series tab. But otherwise, Chris, unless you have anything else you wanted to add, I think that we are calling it a day. All right, sounds like it. Everybody, thank you so much for joining, and we will see you next time.